Hello students, we finished with microsporogenesis. As a result of microsporogenesis, microspores are formed within the microsporangia or the anthers. These microspores later undergo microgametogenesis, and as a result of microgametogenesis, two male gametes and one tube nucleus is formed. Now we talk about megasporogenesis, formation of megaspore within the megasporangium, and afterwards the formation of megagamete within the megagametophyte. We'll be studying first megasporogenesis. Yes, let us talk about megasporogenesis. What it means? It means formation of megaspore within integumented megasporangium. What is that? That is an ovule. It is called megasporogenesis. That means formation of megaspores within the megasporangium. Which is the megasporangium? The ovule is the megasporangium. It has got two integuments usually, the outer integument and the inner integument. Some of the ovules can be unitegument, means only one integument can be there, or some can be bitegument, where two integuments can be there. You can just see the typical flower, this LS through the typical flower. You can see that we have talked about the microsporangia, the anthers, and now we are talking about the megasporangia. It is the ovary. You can see here the pistil, the basal swollen portion is the ovary. You can see the style and the stigma. We're concerned about the lower, this bottom portion, the flask-shaped structure. You have got here the placenta. On the placenta are the ovules attached. You can see here it's very visible. And these ovules are the future seeds. After fertilization, they will become the future seeds. Now let us talk about the development of megaspore. Megasporogenesis. It starts in the megasporangium, the ovule. Now, development of the ovule takes place. You have the formation of integuments. We have, depending upon the type of the number of integuments, we have two types of ovules: unitegument, where only one integument is there, or bitegument, where two integuments are there. Now, we have mostly two integuments in angiosperms. And what happens? That cells are undifferentiated as such. It's a young developing megasporangium. And you will see here what happens, that epidermis is there, outermost layer, and inner to it, in hypodermal region, one cell becomes prominent, dense cytoplasm. It can be distinguished from rest of the cells, and it's called the primary archisporial cell, or we call it the archisporial cell. In some cases, it divides periclinally into two, the primary parietal and the primary sporogenous cell. Or usually, it directly behaves as megaspore mother cell. You can see here, it will undergo first meiotic division and a dyad is formed. Two cells are formed from one single cell, where the reduction division takes place or meiosis takes place. This is followed by second meiotic division or mitosis proper, or we call it equational division. As a result of this, from these two cells, four cells are formed. These four cells are haploid. Primary archisporial cell is diploid, or archisporial cell or megaspore mother cell is diploid. Since it undergoes meiosis, as a result of this, four megaspores are formed, which are haploid. This is the end of megasporogenesis. And as a result of megasporogenesis, four megaspores are formed. In this slide, you can see that from primary archisporial cell, here it is here, primary archisporial cell divides periclinally into two, and primary parietal cell, and this is the sporogenous cell. So this behaves as megaspore mother cell. It undergoes meiosis, and as a result of this, we have got four megaspores which are haploid. So that is the end of megasporogenesis. Megasporogenesis is followed by megagametogenesis. As a result of megasporogenesis, four megaspores are formed. Ovule formation is complete. Integuments cover completely, leaving a space towards the micropylar end called the micropyle. We call it the micropylar end now, and the lower part of the ovule is called the callosal end. So it has got two ends, micropylar end and the callosal end. It is seen that out of the four megaspores, three towards the micropylar end degenerate. They degenerate, and lower one becomes functional megaspore. Now development will take place in this only. It will become the future embryo sac. It has got a haploid nucleus. Depending upon the participation of megaspore, if only one megaspore participates in the formation of embryo sac, we call it as monosporic embryo sac. If two megaspores participate in the formation of embryo sac, we call it bisporic embryo sac. Let us concentrate first on the monosporic type of embryo sac. This nucleus is haploid. A vacuole is formed and the nucleus undergoes mitotic division. The two nuclei are formed as a result of mitotic division and the two nuclei are pushed towards the two ends, or we call the two poles, the micropylar end and the callosal end. The micropylar one after some time undergoes mitosis, as well as the callosal one undergoes mitosis. As a result of mitosis, what happens? Two nuclei at callosal end, two nuclei at the micropylar end can be seen, and this stage of embryo sac is called 4-cell stage embryo sac. It undergoes another mitotic division. As a result of mitotic division, in all 8 nucleate embryo sac is formed, 4 nuclei towards the micropylar end, 4 nuclei towards the callosal end. Now after some time it so happens that these nuclei get organized, cytokinesis takes place and cells are formed. These three form egg apparatus towards the micropylar end, and these three get organized. They will later form the antipodal cells, and the two nuclei from two poles come to lie in the center. Since they are coming from the two poles, we call them the polar nuclei. They are called polar nuclei. All the nuclei are haploid. All the cells even formed are haploid. Towards the micropylar end, the three cells get organized into an egg apparatus. So this egg apparatus comprises of 3 cells, 2 synergids and 1 egg cell. You can see here the female gamete, the megagamete it's called. And towards the center, either the two polar nuclei remain as such, they fertilize when the male nucleus fuses with it at that time, or they fuse to form a diploid secondary nucleus. And towards the callosal end, you can see the 3 antipodal cells. In all, 8 nucleate embryo sac is formed. This is called monosporic 8 nucleate embryo sac, or we also call it as polygonum type of embryo sac. It is also called normal type of embryo sac because it was first observed in polygonum. That's why it's called polygonum type of embryo sac. Since 8 nuclei are there, so we call it also as 8 nucleate embryo sac. With this finishes the completion of the mega-gametogenesis takes place. As a result of mega-gametogenesis, 8 nucleate embryo sac is formed. At last, a big thank you to all my students for patiently listening. Thank you. If you like it, please like it, share it, subscribe it. Thank you.